today's tutorial, we'll focus more on the regulation. Okay, so last time we made, I'm sure those who are there, we discussed the glycolysis pathway itself. Okay, so I'll do a recap of the entire pathway, just in case you miss a class, so that you will know. Then when we are done with the pathway, when I give a recap of the pathway, then what we are going to do, we are going to look at some of the regulatory enzymes and their regulations. Okay, so when we talk about regulation, we are going to be focusing more on the things that inhibit those enzymes and the things that activate those enzymes. And that's where the whole regulation happens. Okay, so now to start the recap, let's start from the glucose molecule itself. So when starting the tutorial last time, we mentioned that glucose is an uncharged molecule. Okay. Then we made mention that this glucose actually cannot cross the cell membrane, which is the plasma membrane. Okay. And then we made mention that the entire glycolysis pathway actually happens in the cytosol. So for this glycolysis to happen, meaning you need to take your glucose molecule to the what? To the cytosol, from the outside to the cytosol. And for it to move to the cytosol, it has to cross the cell membrane or the plasma membrane. And we made mention, since this is an uncharged molecule, it cannot cross the plasma membrane there. So we needed some type of transport to move this glucose molecule from the outside of the cell to the inside of the cell where glycolysis is going to happen. Okay. So now we made mention of some of the transporting mechanism that we use to move our glucose molecule from the outside to the inside. Okay, so today I'll highlight more of those types of transport. Okay, so one of the type of transports, the transporters we use to move the glucose molecule from the outside to the inside is what we call the sodium dependent glucose co-transporter. Okay, so which is the SGLT. So there are two types of SGLT. You have type 1 and SGLT2. These are the types of the transport mechanism used to move the glucose molecule from the outside to the inside. So what this transporter does, it's a co-transporter. All it does is to move the sodium ion. As it is moving the sodium ion, you can see there, you find that even the glucose molecule is also moving inside the cell. Okay, so we are saying this type of transporter is actually an active transport type. Okay, so it uses energy from the sodium potassium ATPase pump. Okay, so it's a secondary active transport type of transport. Okay, so where we are using the ATP, which was generated from the sodium potassium ATPase to move our glucose molecule inside the cell. Okay, so we are saying the SGLT is responsible for the movement of glucose. If we are talking the plasma convoluted tubule of the kidney, is responsible for the movement of 3% of the glucose. Okay, and then it's also responsible for the movement the SLT2. This one is responsible for the movement of 97% of the glucose in the proximal convoluted tube in the kidney. Okay, so that's one type of transport. So we mentioned the SGLT 1 and 2. Okay, so this one we are saying it uses sodium and glucose. So you are moving sodium and glucose in the same direction. Okay, so as you are sodium is entering, also the glucose molecule is going to be entering there. Okay, so that's one type of the transport. Okay, the other types of transport we mentioned is the sodium independent facilitated diffusion type of transport, where the GLUTs actually are falling. So I made mention of the types of GLUT. We have GLUT 1, GLUT 2, GLUT 3. Okay, so and GLUT 4. So for the GLUT 1, GLUT 2, and GLUT 3, these are actually insulin dependent glucose transporters. Okay, so these are insulin independent glucose transporters. Okay, unlike the GLUT4, which is actually insulin dependent transporter. So for this GLUT4 to work, and we made mention that most of the time it's the one we used for the movement of our glucose in adipose, in the heart muscles, and in other types of body organs. Okay, so it's the common one, and this one guy, for this guy, the GLUT4 to work, he actually needs insulin. Okay, so that's what we have. So now what I need you to know is the types of GLUTs where they're actually working. Okay, so you have GLUT1 here, where you're going to find your GLUT1 is in the blood-brain barrier, in the placenta, in the fetal tissues. So you need to know these organs, starting from the red blood cells, the central nervous system, the cornea of the eye, the blood-brain barrier, and the placenta and the fetal tissues. So you should be able to mention some of the organs where GLUT1 is going to work. Okay. Then if you talk about GLUT2, you are going to see it in the liver. Okay, so if you are moving the glucose molecule in the liver, we use GLUT2. Okay, in the kidney, the pancreas as well, for the beta cells, we are actually going to use GLUT2, and also the small intestine. Okay, so you need to know some of these organs where these GLUTs are actually working. Okay, and then we have the GLUT3, which is going to be found in the cells of the central nervous system. You're going to find them in the placenta. Okay, so this is where the GLUT3 is going to work. And take note of this point here. So GLUT1, 2, 3 are actually insulin independent. And then GLUT4 is going to be your insulin dependent. And where the organs you're going to find it is in the adipose, in the heart, and the skeletal muscles. That's where you're going to find this GLUT4. So you should be able to mention the organs where these GLUTs are found. Okay, so that's what we have. So these are the ones which actually help us move our glucose molecule from outside the cell to the inside the cell. Okay, so now we are assuming we've moved our glucose molecule. This is a glucose molecule. It's now inside the cell. Okay, so this is a glucose molecule. It's inside the cell. So let me just say we have glucose there. Okay, so we have glucose. Okay, so this glucose, it's now inside the cell, in the cytosol. Now glycolysis can happen because that's where all the enzymes that usually perform glycolysis are going to be found. Okay, and then you are going to undergo 10 steps for this glycolysis to happen. And what we are trying to achieve here, we are trying to convert this glucose molecule into pyruvate. And we made mention that this will actually happen when you are in a well-fed state. Okay, you are just from taking a meal. Okay, and you want to utilize your glucose so that can be used for different organs as a energy fuel. Okay, so we are going to convert this glucose to pyruvate. As we are converting it, you notice that we'll be generating energy in form of ATPs. That's what we made mention last time. Okay, so that's what we are doing. That's our main target. Okay, so for us to convert this glucose to pyruvate, our final product there, we need to undergo 10 steps. And I made mention that you need to know all those 10 steps and the enzymes which are happening. And I want you to take note of enzymes which are going to work on 3 steps, which are step 1, step 3, and step number 10, the last step. Okay, these are actually regulatory enzymes, and we are going to discuss those ones, because most of the things for this pathway to be stable at all, they will actually target these enzymes. Yes, also other enzymes can be targeted, but these are actually the main regulatory enzymes for your glycolysis pathway. Okay, so make sure you know the enzymes that are working on these steps. These are 3 steps, whether you like it or not. Okay, so you won't do biochemistry without knowing these enzymes that are going to be working on these steps. Okay, so let's continue with the pathway. So we've made mention that we brought our glucose molecule inside the cell, in the cytosol. Then what will be the first step? Okay, so the first step that we are going to do, we are actually going to go convert this glucose to what we call glucose 6-phosphate. Okay, and then you can see here from glucose, we have glucose 6-phosphate. This 6 telling us at portion number 6 of our glucose molecule, we have a phosphate group. We've added the phosphate group. Okay, so now where are we getting that phosphate group? Is actually from ATP. ATP stands for adenyl triphosphate. Okay, so it has 3 phosphate groups. Okay, so we are getting one of the phosphate group and add it to position number 6 of our glucose molecule. That's why we are having glucose 6-phosphate as the product here. Okay, so you notice that ATP here, since we've removed one phosphate, meaning we remain with 2 phosphate group, now it will become ADP, which is adenosine diphosphate. Okay, so that's what we have in there. That's the first step. Now you need to know the enzymes which are going to work here. Actually, so those are going to be regulatory enzymes. Okay, we are saying in most organs like the adipose, the muscle cells, the heart cells, you are actually going to find that the enzyme which is going to work here is going to be hexokinase. Okay, so we are going to have hexokinase enzyme which is going to work here. Okay, and then we made mention that in organs like the liver, okay, the liver, to be specific, the liver, the enzyme which is going to work for this step is called glucokinase. Okay, so glucokinase enzyme. So these are the two enzymes that can work on the first step, depending on the organ type. So I've mentioned in the adipose, in the muscle tissues, in the red blood cells, you are going to find that hexokinase, the one which is going to do that the first step. Okay, so it's the one which is getting the phosphate group from ATP and add it to glucose molecule so that you end up having glucose 6-phosphate. Okay, that's the first step. And then in the liver, glucokinase, the one which is going to do that work. You won't find the hexokinase in the liver. So pay much particular attention to such details so that you don't miss it. So if it's in the liver, just make sure you go for glucokinase. Even if hexokinase is there, make sure you go for glucokinase because you won't find hexokinase in the liver. Okay, so that's the first step. Okay, we'll come back for the regulations for VT enzyme. Okay, so when talking about regulations, we are going to focus more. Okay, so I'm seeing a message here. Is it the are people able to get? Is Joshua the only one not getting me? I don't know. He's saying my network is cutting. Are you able to get properly? Others, are you able to get? Others who are in class, are you able to get? Or it's only him who is not getting?
I don't know if it's just my network here. Maybe it's my network.
Yeah, others, are you able to get me? Yeah, then it's your network. But I'll upload the video on the website. Okay, so try logging out, then you log in. You see if you're going to be getting. I'm sure it's your network. Okay,
All right.
yeah. So continue. Okay, so we are saying we have glucose 6-phosphate here. Okay, that's step number 1. We go to step number 2. Okay, so step number 2, what we are going to do, we are going to convert this glucose 6-phosphate to fructose 6-phosphate. Okay, so what have we done here? So we are moving from glucose to fructose. Okay, so glucose is actually an aldehyde. If we go back to your carbohydrates, you notice that glucose is an aldehyde group. Okay, so it has a functional group at position number 1. Okay, then if you look at fructose, fructose is actually an aldose sugar. Okay, so this is an aldose. So what we've done here, we've just converted an aldehyde to an aldose sugar. Okay, so we've isomerized here. So these are isomers of each other with isomide. Okay, so meaning the enzyme which is going to work there is actually an isomerase enzyme. Okay, so here we are just converting our glucose 6-phosphate to fructose 6-phosphate. Okay, so the enzyme which is going to work there, what was the name of the enzyme? Anyone to give us the name of the enzyme if you are there in class, so that I don't end up giving all the information. Anyone? Hello? Those who are there. Okay, so just make sure you go through whatever we do, just make sure you go through, because we won't be doing anything if you don't go through whatever we are doing. Okay, so now here the enzyme that is going to work, yes, we've made mention is an isomerization reaction. So the enzyme which is working there, it has to be an isomerase enzyme. Okay, so the name of the enzyme is going to be phosphoglucose isomerase enzyme. Okay, so we have phosphoglucose isomerase enzyme. Okay, so that's what we have there. That's the name of the enzyme working there. Okay. So now we move from fructose 6-phosphate. Okay. So this was the second step. This was step number 1. This was step number 2. Now step number 3, what we are going to do, we are going to add another phosphate group to this fructose 6-phosphate. Okay. So we add another phosphate group at position number 1. So meaning we end up having phosphate group at position number 1 and also phosphate group at position number 6. Okay, so we are going to call this phosphate 1,6. Then if 2 phosphates exist in the molecule, we actually use bis to indicate 2. Okay, so we are going to say bis phosphate. Okay, so that's the next molecule. So we are converting fructose 6-phosphate to fructose 1,6 bisphosphate there. Okay. So that's the next step, okay? So the enzyme working there, converting the fructose 6-phosphate to fructose 1,6 bisphosphate is also a regulatory enzyme, okay? And we call it phosphor, okay? So phosphofructokinase 1, okay? So make sure you know this enzyme as well, apart from the enzymes in the first step. Also, this one is also important. It's also a regulatory enzyme. Okay. So now that's the same like hexokinase and glucokinase there. Okay. So what you notice here is that you have one phosphate group here at position number 6, but we've added another phosphate group at position number 1. So meaning what it has done actually, it is to remove one phosphate group from ATP. Okay. Meaning you will remain with ADP there and add it to the fructose 6-phosphate. Then we end up having 2 phosphate group at position number 1 and at position number 6. Okay, so that's the third step there. Okay, so from there you actually go to step number 4. Okay, so for step number 4, you're converting this fructose 1,6 bisphosphate to 2 molecules. Okay, so we are getting one of them to be the aldehyde, and the other one is going to be the aldose sugar as well. Okay, so it's more like fructose, you know, it's a 6-carbon molecule. Okay, so it's a 6-carbon molecule. So 1, 2, 3, 4, 5, 6. So what we are going to do there, we're going to cut there. Okay, so when we cut, we end up having 2 molecules. Okay, and these 2 molecules are going to be having phosphate groups in them. Okay, so meaning we end up having what we call, when we cut this molecule, we're going to end up having 2 molecules. One of them is going to be an aldehyde, and the other one is going to be an aldose sugar. Okay, so we have what we call the dihydroacetone phosphate. Okay, so we have dihydroacetone phosphate, and also what we call glycerolaldehyde phosphate. Okay, so these are the 2 molecules. We've split down our fructose into 2. Okay, so we end up having dihydroacetone phosphate and glycerolaldehyde phosphate there. Okay, so that's the 4th step. Okay, and the enzyme working there, so you can see that we are forming aldose sugars there. Okay, so we're getting aldose sugars. Okay, so the enzyme working there is an aldolase enzyme. Okay, so this is aldolase enzyme working there. Okay, so it's just cutting the molecule into two. Okay, so now for glycolysis to proceed, it doesn't work with the glycelo and dihydroacetone phosphate, this one. Okay, so it doesn't work with dihydroacetone phosphate. Okay, it actually works with the glyceroldehyde phosphate. Okay, so what we need to do is to convert this dihydroacetone phosphate back to glyceroldehyde phosphate. Okay, so meaning here what is going to happen is that this glyceroldehyde phosphate, which has been formed here, it will be brought the way it is. Okay, so we still have glyceroldehyde phosphate there. Then this one, what we are going to do, we are going to convert it to glyceroldehyde phosphate there. So we end up having two glyceroldehyde phosphate. Okay, so meaning we will convert this dihydroacetone phosphate to glyceroldehyde phosphate. Okay, so the enzyme that is working there is, so this is an isomerization reaction as well. Okay, so you can see these are 3 carbon molecules. What we've just done here is to move the functional group. Okay, so the functional group for this one, this is an aldehyde, this is a ketone. So we've converted the ketone to an aldehyde. So it has just moved a functional group from position number 2 to position number 1. Okay, so meaning the enzyme that is going to have work there, since it's a triose sugar, we're going to say triose, okay, triose isomerase enzyme. That's the name of the enzyme which is going to work there. Okay, another type of enzyme, just in case if triose isomerase enzyme is not there, another enzyme which can work there can be called glyceroldehyde phosphate hydrogenase enzyme. Okay, so you can have glycerol, okay, aldehyde phosphate. Okay, so glyceroldehyde phosphate dehydrogenase. Okay, or not dehydrogenase, you can have hydrogenase. Okay, so hydrogenase enzyme. That's the name of the other enzyme that can work here, apart from the triose isomerase enzyme. Okay, so that's it. So meaning at the end of step 5, you have 2 glyceroldehyde phosphate here molecules. Okay, so I hope we are following up to here. Are we clear up to step number 5? So now the entire step from step number 1 to step number 5, this is called the energy investing phase. Okay, so this is the energy investing phase. As you can see in these steps, we are actually using ATP to form what? ADP there. Okay, we are utilizing ADP, okay, to get ATP there. Okay, so this is the investing phase. Okay, so we also have the phase 2 there, where we are actually going to generate, okay, our energy there. Okay, so meaning here, as we are moving, we are actually going to be getting energy in form of ATP. Okay, so let's do that. So we go to step number 6. Okay, so step number 6, what we are going to do to glyceroldehyde phosphate. Okay, so meaning whatever is happening here is also happening in the other side. Okay, so I'll just be using one side so that I have enough space. Okay, so meaning whatever is happening here is also happening in the other side. So whatever you'll be producing twice here. Okay, so the first step in the energy generating phase, which is step number 6 of glycolysis, the first thing we are going to do is convert glyceroldehyde phosphate to what we call 1,3 bisphosphate bisphosphoglycerate. Okay, so now 1,6 bisphosphoglycerate. So here it means that we add another phosphate group at position number 1. Remember this glyceroldehyde phosphate, it's a 3 carbon molecule. It's having a phosphate group at position number 3. Okay, now we add another phosphate group. No wonder we're having 2 phosphate group. So we end up having a molecule called 1,3 phosphor. So BC. This means 2 phosphate group, isn't it? So we end up having 1,3 bisphosphor glycerate molecule. Okay, so we have 1,3 bis phosphoglycerate. Okay, there. That's the next step. That's steps number 6. Okay, so there, this is where the first redox reaction is going to happen here. So for the redox reaction, what we are going to do, we are going to utilize ADP plus. Okay, so we have NAD plus there, NAD plus there. We are going to reduce it to NADH. Okay, so that's a redox reaction. That's the first step, step number 6. Okay, so we've converted our NAD to NADH there. Okay, so this is a dehydrogenase enzyme working here. Whenever you see NAD and NADH, just know that the enzyme is going to work there. It's a dehydrogenase enzyme. Okay, so the enzyme, the name of the enzyme is going to be glycolysis dehydrogenase enzyme. Okay, so that's the name of the enzyme that is going to work there. Okay, so we move. Okay, so this is glycolysis dehydrogenase enzyme, which is going to work at step number 6. Okay, so we move, we go to step number 7. Okay, so for step number 7, we have 1,3 bisphosphoglycerate. Okay, so what we are going to do here, we are going to remove one phosphate group from this 1,3 bisphosphate. Okay, so we remove one phosphate group. When we remove one phosphate group, meaning only remain, so the phosphate group we are removing is at position number 1. So when we remove that one phosphate group, meaning we remain with only one phosphate group at position number 3. Okay, and the name of the molecule which is going to be formed, so we removed one phosphate, meaning we are only remaining with the phosphate at position number 3. So it's going to be called 3-phosphoglycerate. Okay, this was called 1,3 bisphosphoglycerate, but here the bis is going because we have only remained with one phosphate group plus the glycerate. So we are going to call it 3-phosphoglycerate. Okay, so this is 3-phosphoglycerate, which is going to be formed there. Okay, so that's step number 6. Okay, so step number 6. Okay, so now when we remove that phosphate group, okay, when we remove that phosphate group, where are we taking it? Okay, so now remember ADP. Okay, so we can get one phosphate group from 1,3 bisphosphate. Okay, so we get our one phosphate group here, we add it to ADP. Okay, so this ADP now we have 3 phosphate group, which is the ATP here, which has been formed. That's the first ATP which are generating in glycolysis. Okay, so now if we generate one ATP, meaning even the other side we are generating another ATP. So we have 2 ATPs so far at position number 6, at step number 6. We have ATPs which we've generated. Even the NADH which we've generated here, you notice that this side we've generated one and the other side we've generated one, since we are the 2-glyceraldehyde phosphate. Okay, so that's what we are having. So we have 2 ATPs which are going to be generated at that step. Okay, so now from there we can move. Okay, so this was step number 6. We can go to step number 7, where now we are converting our 3-phosphoglycerate to 2-phosphoglycerate. Okay, so we are isomerizing here. So the name of the enzyme is going to be amylase. When there is isomerization reaction, it's either an isomerase enzyme which is going to work or a mutase. So we are actually isomerizing our molecule. Okay, so we are moving our phosphate group from position number 3 to position number 2. So we end up having 2-phosphoglycerate molecule there. Okay, so the enzyme which is going to work there is going to be called the phosphoglycerate mutase enzyme. So it's a mutase here. So it's a mutase enzyme because there's an isomerization reaction. Okay, so we have phosphoglycerate mutase enzyme working there. Okay, so now from there, what we are going to do, okay, so what we are going to do from here, we are going to move from 2-phosphoglycerate. Okay, so what we are going to do, we are going to actually convert this 2-phosphoglycerate. We made mention we are converting it to PEP. Okay, so PEP. Okay, so that is actually done by the enzyme. Okay, so that is done by enzyme enolase. Okay, so we are converting 2-phosphoglycerate to PEP, which is phosphoenolpyruvate. Okay, so this is phosphoenolpyruvate by the enzyme enolase. Okay, then from there we can now convert our PEP to pyruvate. Okay, so we can convert our PEP to pyruvate. Okay, so here again, what we are going to do, we are going to remove the remaining phosphate group from the PEP there. Okay, so we remove that phosphate group, we add it to ATP, meaning we are generating another ATP. So meaning 2 more ATPs have been generated there. Okay, so that's what we have. And the end product which we made mention was the 2-pyruvate being formed there. Okay, so are we clear on the entire pathway? Are we clear there on the entire pathway? Before I go to the regulations. So I'll be fast when talking about regulations. Are we clear on the pathway, the entire pathway? Okay, yeah. So at the end of the glycolysis, you also need to take note that from one glucose molecule, we have one glucose molecule, you actually end up obtaining 2 ATPs. Okay, so you notice that why we are getting 2 ATPs is because, okay, it's because here in the investment phase, we actually used 2 ATPs. So from the 4 ATPs here, which we produce, we actually also consider the 2 which has been utilized. So we will remove the 2. So meaning the entire glycolysis will obtain 2 ATPs, okay? And then the other thing, you also obtain 2 NADH. Okay, so 2 NADH, which are obtained at portion number 6 there. That's those are the final, and 2-pyruvate. Those are the end products for the entire glycolysis pathway. Okay, so now in terms of regulations, we are actually targeting those regulatory enzymes. We've remembered the ones which are working at 1, 3, and 10. Okay, which were the first one, it was hexokinase or actually glucokinase. And then here at portion number 3 was phosphofructokinase 1, and at portion number 10, which is pyruvate kinase enzyme. Okay, so those are the enzymes we're actually targeting here. So what you need to know about regulation is what inhibits and what activates that enzyme. Okay, so I'll go straight for the activation. Okay, so for hexokinase, what inhibits it is actually the products itself. Okay, so hexokinase is working at portion number 1, where we're converting our glucose to glucose 6-phosphate, isn't it? So the glucose 6-phosphate, which is being produced by that enzyme, can actually act as an inhibitor for the enzyme itself. So if you're having more of this glucose 6-phosphate, meaning it's going to inhibit your enzyme hexokinase. Okay, so that's one of the regulations we need to know about this enzyme. Okay, so in terms of glucokinase, okay, so for glucokinase, we are saying that this glucokinase is actually going to be activated by insulin. Okay, so insulin is the one which actually activates glucokinase. Okay, so insulin is going to be secreted when you are just from taking a meal. Okay, so you are going to get, after some few hours after taking a meal, you have this hormone insulin which is going to be secreted and is actually going to activate this enzyme glucokinase to start doing its work. So insulin is actually the activator for this glucokinase enzyme. Okay, so that's what you have. So unlike hexokinase, this glucokinase cannot be inhibited by the product, which is glucose 6-phosphate. Okay, at the physiological concentration. Okay, so that's another thing you need to take note of. So take note of the things that activate the enzymes and inhibit the enzymes when it comes to regulations. And these are the questions where you'll be asked most. Okay, so for phosphofructokinase, the one which was working at step number 3, okay, So the things that inhibit phosphofructokinase, allosterically, you can have ATP and citrate. These are two inhibitors for phosphofructokinase 1, and it can be activated by high concentration of AMP. Okay, so that's what we have. Okay, also fructose 2,6 bisphosphate via the CAMP pathway is also able to activate phosphofructokinase 1 as well. Okay, so that's what we have. Those are the regulations for phosphofructokinase 1 enzyme. Okay, so hyperglycemia, it means high glucose levels, are inhibitors of glycerate 3-phosphate dehydrogenase enzyme. Okay, so this enzyme is the enzyme working at step number 6. Okay, where we are getting NADH. Okay, so the enzyme, that enzyme can be actually inhibited by high concentration of glucose in the body, which is hyperglycemia. Okay, so that's another thing you need to take note of. Okay, away from the enzymes, okay, we can have arsenic poisoning. This one can actually inhibit your glycolysis without actually inhibiting the enzymes themselves that are working on glycolysis. So it has other ways in which it can actually inhibit the entire pathway without touching those enzymes which are working on the pathway. Okay, so how this happens, it's you can have arsenic which will prevent ATP and ADP production without inhibiting the entire pathway itself, like I've mentioned. Okay, so it will interact with cysteine SH group of glycerate 3-phosphate dehydrogenase enzyme. Okay, so when it interacts with the cysteine SH group of glyceroldehyde 3-phosphate dehydrogenase enzyme, which is working at step number 6, it will form a complex, okay, which is 1-arseno-3-phosphoglycerate complex there. Okay, so this will spontaneously hydrolyze to form what we call 3-phosphoglycerate. Okay, so by bypassing the synthesis of 1,3 bisphosphate, ATP is not formed by phosphoglycerate kinase. Okay, so that's how this acetane will actually inhibit glycolysis without actually inhibiting any of the enzymes in that pathway. Okay, so that's what we have. That's another regulations you can have. Okay, so this is a summary of the entire regulations. Okay, in terms of the enzymes, here we have hexokinase. Okay, so hexokinase, it has high Km. Okay, so we need to know some of the things that can inhibit it is the product itself, which is glucose 6-phosphate. Okay. Okay, and then we have said you can have glucokinase, which can be activated by insulin. Okay, then we have the enzyme working here, which is phosphofructokinase 1. Okay, so it can be activated by AMP, fructose 2,6 bisphosphate. Then it can be inhibited by ATP and citrate. Then for the last one, the last enzyme, which is pyruvate kinase, can actually be inhibited by phosphorylation via the CAMP pathway and alanine. Okay, and then it can be activated by fructose 1,6 bisphosphate. Okay, so these are some of the regulations for your glycolysis pathway. We have a question before we end our meeting. Is everything okay? Are we having a question here? Do we have a question? Okay, so if there's no question, we'll end. Yeah, so we end here. Thank you so much.